to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're looking at the basic structure of mononucleotides and the structures of DNA and RNA. You should also know how complementary base pairing and hydrogen bonding between two complementary strands are involved in the formation of the DNA double helix. So a nucleotide is a structure that contains a pentose sugar, a phosphate group, and a nitrogenous base. A single nucleotide is considered a monomer. The sugar base differs depending on the structure built. So for example, in DNA, the sugar is deoxyribose. The bases in DNA are A, which is adenine, T, which is thymine, C, which is cytosine, and G, which is guanine. In RNA, this sugar is a ribose. The bases in RNA are the same except T, thymine, is replaced with U, which is uracil. So what are purines and pyrimidines? So purines are the bases with the double ring structure, and this will be adenine, A, and guanine, which is G. And you should know the names because in GCSE you were used to just writing the letters. But at A level, you need to know the full names of the bases. Pyrimidines are the bases with a single ring structure. So this will be cytosine, thymine, and uracil if it's the case of RNA, and it will be thymine if it's DNA. Nucleotides in a DNA structure are joined together in a condensation reaction, and the bond that forms as a result of this condensation reaction is called the phosphodiester bond. So, two antiparallel polynucleotide strands are held together by complementary base pairing. These strands are held together by complementary base pairing such that A and T form 2 hydrogen bonds and G and C form 3 hydrogen bonds. The two strands twist to give a double helix structure. So now we're looking at the RNA structure. So RNA, unlike DNA, is a single-stranded structure and contains a ribose sugar and uracil instead of thymine. And examples of RNA are mRNA, tRNA, and rRNA. So now we're looking at the complementary base pairing and its roles. This ensures that each base binds to the correct base pair. And that ensures that the antiparallel structure is maintained, where one strand runs from 5' to 3' and the other strand runs from 3' to 5'. So the 5' end has a phosphate group attached to the 5th carbon of the sugar, while the 3' end has the hydroxyl group attached to the 3rd group. This ensures that all DNA replication and transcription that happens is accurate, and this minimizes the risk of mutations or incorrect proteins being produced. So that is all from Professor SNAB today. Hope you found this video useful. If you have any questions, let me know in the comments below and I will get back to them as soon as possible. And don't forget to like, share, and subscribe.